this time, Chad is around with the microphone, and we have Brian available and Joel. And of course, Michael is sitting here with us. And so we'll start off with Mr. Becker.
I have a question for Joel.
Okay.
Back to where you showed and I would assume they're mostly bacteria in the seed, and you were suggesting that it may actually inoculate the soil as it germinates and gets started. And what I'm wondering is if these bacteria become sort of a characteristic microbiome that the plant takes along with it. And so there is a symbiotic relationship between those two that goes on for generations.
Yes, that is exactly correct. That is what happens because each generation will send the same, it ends up being the same microbial species that go to the seed, each generation for that particular plant species. So this is part of then what's called also a core microbiome, is that it's the microbes that are associated with a plant species irrespective of the soil type, of the climate, of the, et cetera, all of those other variations of microbes that can exist around the world and in different soil types. But the core microbiome is unique to that plant species, and it remains constant along all of those variations. So yes, you're quite right. So this is called vertical transfer of the seeds, of the microbes through generations, where the next generation is acquiring those microbes. But the seed can also acquire microbes horizontally, which simply means, from the soil and other ones in that local native environment. So you can get kind of this horizontal transfer or environmental transfer, but the vertical transfer is really direct from the mother plant, and that is constant through generations. And I think that that's also, this is one of the reasons why saving your seed, I think, is actually a really important strategy, because the more you grow on your specific soil types with your unique soil microbiome, and you get a certain expression of the plant in that environment, which is then also translated through into the seed, the more you save that seed and regrow that in your specific kind of microclimate and soil type, you're actually strengthening and fostering some of those interactions that take place. So I'm a big fan of seed saving. I think that when you buy seed that comes from maybe a very different soil type or a very different environment, you might have some of those horizontally transferred microbes that aren't necessarily the best for your soil type or the best to thrive. And as I pointed out at the end there, as we now know, microbes themselves can induce changes in the plant. Microbes can change the flowering time, change various plant phenotypic traits. So a variety that was bred and selected for in a different soil type with a different microbiome, the plant expression of that could be unique to that soil microbiome. And you put that same variety in a different soil microbiome, it may not actually behave the same. The plants may not respond the same. So yes, these things are carried through the generations, and I view this as a reason as to, I think, why seed saving is a potential valuable tool.
Other questions for the group in the back? Yes, Scott.
Older varieties of say wheat might have different exudates, and older varieties of wheat say might have different exudates than newer varieties, right? Do we have evidence for that?
Yeah, there would be, for sure. I can't say I've seen that specifically, but yes, there would be differences. I mean, we know that even modern differences amongst modern varieties, there are differences. And that's, as you know, like different varieties might be whatever, insect or disease that level of disease resistance, let's use disease resistance as an example, it often is just a couple of genes that are different in some of those varieties that then give them that resistance against the disease. So similarly, a couple of genes different from variety to variety means that the behavior of those different genes can induce differences in the root exudates. So I can't say I've seen a paper specifically, but for sure, older varieties would contain some unique and different exudates than modern varieties, for sure.
Do you know if any studies ever show that on the same plant, some roots for the plant produces some exudates and other roots produce other exudates?
Yes, that does happen. I'll tell you what, I'll even quickly go back to this example and show you. Here we go, sorry. Hopefully you can see that now. So yes, that does happen because some of the root exudates are more active in the root tips, for example. That is where a lot of the root exudates go, but you also get some other root exudates a bit further back. And so this was the picture I want to show you. The phenomenon that you're talking about is right here present in the soybean. You can see that some parts of the root systems are a little more on the neutral side, and other parts of the legume root systems are a little more on the acidic side. So yes, it is possible that different parts of the root systems are releasing slightly different exudate composition, yes.
Other questions? Yes, in the back there.
In regards to cover crops and trying to get the best nutrient value out of the cover crops, what's the best way to go about? Is it to shallow incorporate cover crops, desiccate them with a herbicide? Because when we go and we test cover crops and you say there's, whatever, just say 80 pounds of nitrogen, 30 pounds of phos, and whatever else, we're cutting the biomass off, sending it in, that's what's in the biomass. But how is that getting back into the soil without making contact to the soil?
Great question. So, we had this big discussion about roots, not shoots. The reason that roots are so much more valuable and make such a greater contribution to soil organic matter is no big secret or trick. It's simply where they're located. They are located already down in the soil where the microbes are, where they can be processed. Now, shoots, of course, are accumulating on the surface, and that makes them therefore more prone to oxidative losses. Here I'm talking about carbon just for a minute to come to your question in a second. So that is why shoots make a less of a contribution, is because of their physical location on the soil surface. Now, you can increase the contribution of shoots towards soil organic matter, yes, by incorporation. If you incorporate that shoot material, for example, shallow incorporation, if you incorporate that in to optimize more residue soil contact, then yes, it turns out that shoots begin to make a bit of a bigger contribution to soil organic matter than otherwise. However, of course, that comes at a cost. That disturbance, that soil cultivation is then breaking apart. The thing that is concerning the most is that as we disturb the soil, we are breaking apart those aggregates that I also talked about. So, and aggregates, we have the chemical stabilization through the mineral surfaces, and we have the physical stabilization through the aggregates. When you disturb the soil, it is particularly the aggregates that are vulnerable. They are the ones that break apart, and therefore any potential carbon is exposed and potentially lost. So that action of integrating to trying to get more out of shoots and integrating that into the soil surface, you get a win and you get a loss. It's kind of an overall neutral position in my view. So I think much more preferable is to leave the shoots on the surface. Okay, and then two things. I think shoots are better to go through an animal digestion. And that animal could be, livestock, cattle. I can see Brian's giving some thumbs up here. Those shoots above the shoots, the above ground residue, put that stuff through an animal digestion. Now, not just livestock, I would also include earthworms in that category. So, of course, the worms coming up and them taking the shoots and burrowing those down, and those worms putting them through their digestion is also the same. So this is the thing. I think that shoots can make a bigger contribution to soil organic matter, providing we've got worms, which of course tillage is not the friend of worms, to let then the worms bring that shoot material down and put it through their stomachs, their digestion, okay, or livestock alternatively, and to get the real benefits kind of that way. So that is what I think is the optimum scenario. Now, sorry, that was a long way to give that analogy to come to your question, to say that nutrients, on the other hand, unlike carbon-based compounds, the organic matter, which can be lost as CO2, the nutrients, most of the nutrients, do not form gaseous forms that will drift away. So even if residues sit on the surface for a long time, you'll have a great deal of the nutrients will still remain there. They're just going to be unlocked very slowly and released very slowly. Again, sped up if you graze it with livestock, sped up if you graze it with earthworms. You will speed up that process, but for the most part, the nutrients themselves staying on the surface is okay. You'll just get a slower breakdown and a slower release, and then a slower leaching kind of down into the soil.
I feel like it's kind of a catch-22, whether you use herbicide or just a shallow incorporation of tillage. Has there ever been any studies on either or, as far as which one's more beneficial or more detrimental?
Yeah, this is a tough question, isn't it? I don't know the answer to this. We know that, for example, glyphosate has some chelation capacity. It does bind to certain nutrients and immobilize them. Yeah, so I'm sure other herbicides and things would have some similar mechanisms potentially. But hard to say whether that would be like long term. That could be more of a temporary, transient effect. Microbes are very good at breaking down chemicals as well. They can break down and detoxify these things. So at that point in time, maybe the nutrients would be released. But I know we all have also a big fear of disturbance. I'm not a total anti-tillage guy. I think small amounts are okay. But again, keeping it shallow, as to your question, I wouldn't say no to that. I think that, deep plowing, is more of a big no-no, but maybe some strategic and shallow incorporation, I'm not opposed to. I think that's providing you're incorporating some food, some residue. I don't support shallow tillage if you're just doing it for bare fallow and there's no plant material kind of getting integrated. You're not feeding the soil at the same time. It's just, you're just disturbing the soil for the sake of it. That makes no sense. But strategic disturbance to incorporate some food. I'm not super anti that either, as long as it's kept shallow and maybe some strip till or something to hedge your bets there. But no, I can't say I've seen anything specifically on nutrient release of one versus the other.
I have a question for you, Brian. Can you explain a little bit about your use of full season cover crops and from an economic standpoint, them not working for you? And I guess my question is, are you figuring in the winter graze and that, or did I may have interpreted that wrong as well? You want to elaborate on that?
Yeah, absolutely. I didn't feel like I had enough time to really thoroughly explain that. And so about 4 years ago, I dedicated 2/4 sections of property right up and close to our headquarter operation there, that I decided I wanted to do a zero carbon removal for like 4 to 5 years. And in that process, of course, you're planting a full season cover crop as a cash crop, and you're putting livestock on it, and they're doing all the removal. That part of it was in and of itself, over the course of the 4 years, very successful. 2 years ago, in fact, we had a group of spayed heifers and steers on those 2/4 sections, and we rotated it, intensely grazed it as if it were a pasture. So we cordoned it off in smaller paddocks and moved them every 4 or 5 days. And we ended up with about a 3.3 pound gain on those critters. So that was in and of itself very successful. But when you look at, there wasn't anything hardly left though for any winter grazing because it was a full season and we were grazing it pretty intensely. And we didn't take it all down, but we did do some grazing on it that winter, but not enough to really count as enough, maybe I think 20 days that we had off that. But when you look at that versus a wheat crop, for example, where you've got an 80 or a 90 bushel wheat crop and you market that through a certified seed system, where you're value adding that and then putting a cover crop on it and then putting a bunch of return lease bulls that have gone out on a $1,500 lease and then come back and get reconditioned on that. When you look at it in that scope, the full season cover crop in and of itself doesn't compete with that. It still works, but it didn't compete with going through a full term cover crop graze scenario in our system. But I have to emphasize that it still, from an economic standpoint, worked. Now, a year like 2021, it probably wouldn't have worked because there just simply wasn't enough moisture to provide enough forage for an intense amount of animals. It would have been less than half of what we could carry the previous 3 or 4 years. So each year is going to pose a different opportunity and a different threat. But again, we have to look at things from a broad perspective of economics. And when we look at it from a systems approach, it's not as good as if it were cropped first.
Thank you for that explanation. Any other questions or comments that anyone would have? Dave?
Joel, you talked about a lot of the complexities, and I guess I wonder about how you manage these things. I think you talked about the principles helping kind of lay out the general guidance, but things like diagnostics within the year. You mentioned SAP tests, tissue tests. I guess just some general comments on how you manage these systems with all that complexity.
Yeah, and of course, trying to measure and manage the soil biology is also, okay, there's interest in this and there's some labs kind of doing this, and yeah, I think there can be some benefit to this. But yeah, it's so complex and we know so little that there is a question mark over how valuable or useful some biological testing is. I think that is changing and will change. I think it will become something more valuable and useful over time. And again, as I mentioned, we have, again, I'm not opposed to microscopy, but it's a bit of a basic kind of test. It's kind of crude in its means, but sure, it's better than nothing. And then we have the genetic tools, which are so advanced, but they're just kind of way too academic at the moment. They're not applied enough. So we're in this tricky middle ground position where we clearly have some way better tools, but we don't yet have maybe full practical translation of those. So we're in this annoying kind of middle ground. But is microscopy a useful tool, especially for on-farm, looking at composts or looking at compost extracts and some of these kind of basic things? Yeah, I think they're still very good tools for that purpose. So I think that, yeah, the biology one is just a tricky one. So beyond that, yeah, I am a fan of SAP, either SAP or tissue tests. I don't mind either. I think both are good. And I'm a fan of those because there's also a lot of complexities with soil chemistry testing. We know that pretty well. There's good stuff there, but there's a lot of nuance to that in terms of testing what's available versus the totals. I'm a fan of doing kind of totals tests and looking what that total pool of nutrients is. I think that really motivates you on an economic level to realize the reserves that you do have in the soil and then focus on the biological strategies to kind of liberate that. But where I kind of think foliar or plant analysis is good is to then use those foliar applications for really targeted inputs to really help drive plant health or drive production which at the same time keeps some of that nutrient burden out of the soil. Of course, we know that if nitrogen builds up too much, nitrogen fixation shuts down. If there's too much phosphorus, mycorrhizal associations shut down. So there's plenty of examples. We know that happens. So wherever we can, trying to limit the amount of excess fertility entering the soil is really key. And that's why I am a big fan of the foliar approach. Small amounts, it's going into the canopy, not so much on the soil, but just enough to prime that photosynthetic pump. And where that works is, that small amount of nutrient that you applied, it's insignificant, but what it's really doing is priming the pump so that then we prime root exudates, and it is those exudates that then drive nutrient cycling through the soil biology, which then delivers the nutrition back. And that amount of nutrients that you get back from the soil through that indirect activation of the soil microbiome, that amount of nutrients is far greater than that little amount that you put on the foliar spray on top. And that's why I think there's a lot of opportunities there with really targeted foliar spray. So and I think that when you're doing those plant analyses, you're asking the plant directly. You're going straight to the horse's mouth. It's all good to look at the soil and see what's there or not or available and this kind of nuance, but really the key is, well, what does the plant actually have in it at this moment? It might be there, but is it accessing it? Is it accessing it? Or, so that's why I think that the plant is more going direct to the plant, going to the horse's mouth, and then you can tweak your management process from there. So yeah, I'm a big fan of plant analysis, but sure, I would still do soil chemistry testing as a broader strategy as well.
I have one quick question for Michael. Take me through your 44-inch corn process. How long have you been doing it? Have you got a yield drag data that you can share or increase? And are you moving over, or what's your process? Because obviously we're playing with some 60-inch corn at the Minokan farm. So last year yeah.
So last year was a drought. I don't know if that's a fair comparison or not, but we were probably 10% down from the neighborhood, something like that, 5 to 10, off of a full tillage, full high maintenance version. Now our cost structure was greater than 10% less, so I think it's a win. It gives us a lot of room for diversity. With 44-inch rows, we get a little sunlight, and our cover crop grows, as you know, on 60. When you try and do that on 22, it's pretty tough. What was the rest of your question?
It's a sunlight game, right?
Yes, very much.
So are you planning on moving over then? You're using GPS technology, and then you're going to plant into the cover? Or?
So we only do about 10% of our total crop to corn.
Right.
And that's on a big year. Sometimes it's less.
Oh, okay.
So there'll be a multi-species interseeded in everything we got done, which is over half of it. That will have rye that came through the winter. So next spring we'll seed soybeans into that more than likely. Although a half section of it that's in the home section will probably get cattle grazed on it if the timing works out before we seed the soybeans. We might wait a little bit to seed the soybeans just to get a quick graze across that.
Okay.
Economics, I'm not sure yet.
Right. Okay. Well, before we wrap up, is there any other? Oh, neat. Well, hello. Hold on, please. There you go.
On your native prairie, do you have much Kentucky bluegrass expanding?
I assume that question's for me.
Yeah.
No. In fact, we're through the intense management that we've employed the last several years, we've actually gotten rid of areas of Kentucky blue, and which is good. And it's bringing back the more native species of the gammas and some of the smaller reds and so forth. And so it's really been a rewarding for us to go through that intense management process and learn each year the nuances that the native prairie system brings to us. Did
So
that
you
answer
are-
your question?
Yes, but so then you are grazing probably early in the spring and sometimes winter.
Well, we have an intensive artificial insemination program on our cows, and it's about a 5 to 6 week process from start to finish from our heifers all the way through our mature cow herd. And they all hit grass about the same time, and that's about the end of May. So there are some paddocks that we'd like to hit earlier than that, but because of the nature of our management, AI management, they don't get out there as soon as they could. But we try and make up for that just by intense movements every 4 to 5 days.
Thank you.
Neil had a question, and then we'll take it to Michael, and then we'll wrap it up.
This question is for Brian. And have you done any economics on co-cropping with a cool season cereal grain and a cool season legume like peas, for example?
Well, yes. Well, we've been raising peas and oats, for example, and triticale and oats for many, many years, since the early 1990s. And we see usually about a ton to a ton and a half advantage as a forage crop with the companions. We do not, however, use companions in the cereal grain side because most of our acres are devoted to seed production. And so that limits us through the inspection process of having, which I'm trying to change, by the way. We're trying to get them to allow certain other species in that mix, or at least growing with it during the inspection processes. So we have not, I've tried some clover and different things on non-feed acres, but I've had very limited success in getting that established quick enough. We get pretty hot pretty early. As you can imagine, in southern South Dakota, we can get, in fact, our spring wheat's already in the ground, guys. We got done planting spring wheat Friday. So we have to plant early to make spring wheat work, and getting clovers or anything like that to work well with it are very difficult. Now, forage crops are different. So we're going to try experimenting with a field pea like a cow pea, or a cool season pea with the, not a cow pea, a cool season pea with the forage winter wheat this spring, and see if we can get some success with that. We've also done 60-inch corn. We're going to move away from that because we have absolutely no economic gain from that. And Daryl said it best when he said it's a photo game. You know, you're trying to capture as much sunlight as possible, and we just didn't see an advantage with the 60-inch corn.
Michael, go ahead.
Can you hear me? There we go. Brian, you had an acronym SWOT, I believe it was. Can you repeat what that was?
SWOT, SWOT, strengths, weaknesses, opportunities, and threats. It's a process that I've been through a number of times on different boards that I've been on, but we employed that process on our operation about 10 years ago. And it's pretty in-depth depending on how you do it, but it forces you to really sit down and analyze your whole business, and it also helps you understand your personalities within your business. And that's probably more important than anything, is you learn how to deal with each other.
Can you repeat that one more
Brian,
time?
just go ahead, and it's SWOT, it's strengths, weaknesses.
Weaknesses, opportunities, and threats.
Thank you. I think that's a great one.
Well, I think with that, it's 4:30, folks. Just a couple things. I'd like to thank you all for your participation today and hanging in there with us. The speakers were very, very good. I think they did a great job. Appreciate you guys being part of it today, Brian and Michael and Joel. And we look forward to seeing you guys in person sometime, and the opportunity to come up to the Menoken Farm or Burleigh County or wherever we'll see you down the road. And again, appreciate all that you guys do. And let's give them one more round of applause.